We're going to start our program, and I have the great pleasure of introducing a colleague and friend of mine, Professor Ahad Rahim, who's a professor of translational neuroscience at University College London. And he will be talking about our efforts into a gene therapy approach for PLA2G6 associated neurodegeneration. Ahad.
So thank you very much, Manju, for the very kind introduction. And thank you to everybody, NBIA committee, for the invitation to talk to you all. I wanted to start off actually just by reiterating something that Michelle mentioned, which is the importance of the family organizations in supporting researchers. I've been involved in developing gene therapy for maybe 8 or 9 different indications, different diseases. And I have to say that invariably across the board, the family associations have played an absolutely critical role in getting those programs started, but also play an important role in actually starting clinical trials. So my heartfelt thanks to NBIA Association. So I'm going to be talking to you today about a program of work that we've been working on at UCL for a number of years now. And this is to develop a gene therapy program for infantile neuroaxonal dystrophy. So INAD is a type of neurodegeneration. It's one of the different diseases that comes under the brain iron accumulation umbrella. And it's caused by a mutation in a gene called PLA2G6. Now, that gene makes a protein which is involved in a number of important functions that take place in our cells. And I'll come back to that in a moment. But INAD is the most severe form of these PLA2G6 associated diseases. And clinically, I mean, I think Professor Manju Kurian is going to tell you a little bit more about the clinical details of the disease, but to just touch upon them and just to re-emphasize the importance and severity of this disease, is that at birth, patients look normal and they develop normally during early stages, but you start to notice these progressive motor and cognitive decline in the patients, and there's this loss of neurodevelopmental skills. And so it's a progressive disease, and kind of the mean age of death is around 9.1 years of age. Unfortunately, there is no major disease-modifying treatment available for patients at the moment, and it's a sad state of affairs that the only treatment that's available is palliative care. Now, this is the kind of driving the motivation for us to develop this kind of gene therapy approach for treating INAD. So before I go into the nuts and bolts of gene therapy and the program that we've developed, I just want to take a step back and just go through the kind of fundamental biology of how mutations in genes cause diseases. And the reason I want to do this is because if I don't get this bit right with you, I'll lose you right from the beginning, and I don't want to lose you right at the beginning. I want you to follow me through the journey that we've been through. So here we've got a cell on the left, and you can see at the top you've got DNA, and it goes through this process which results in a protein. Now, in a condition like INAD, you end up with a mutation in the DNA or a mutation in the gene, which in the case of INAD is the PLA2G6 gene, which I mentioned earlier. And as a result of that, you end up with a protein that doesn't do its job. And when that protein doesn't do its job, you end up with a genetic disease, which in this case is INAD. Okay. So again, you've got your gene. Gene is mutated, it's got a problem. The protein it produces is then going to have a problem, and therefore you'll develop INAD disease or genetic disease. So what is this gene therapy that I'm going to be talking to you about, but you maybe have heard yourselves about through your own reading, through what you heard in the news, through other parents of patients, or through other disease organizations. And there are a number of different ways of defining gene therapy. I actually really like Wikipedia's definition. So Wikipedia says that gene therapy, also called human gene transfer, is the therapeutic delivery of nucleic acid into patient cells as a drug to treat disease. Okay. So the nucleic acid we're talking about here is the gene, PLA2G6 gene, and we want to deliver that into the cells of patients. So, okay, makes sense. The idea is to take a healthy version of PLA2G6 and deliver that into the cells of patients to compensate for the defective gene that's there. And that sounds very easy in principle, but actually it's a bit more difficult than that. And the reason it's so difficult is because the cells of our bodies have lots and lots of barriers in place to prevent genes coming into our cells and corrupting our own genomic DNA. So here we've got a picture of a cell, and around the outside you can see there's this thing called a cell wall. And then when you get inside the cell, you've got all these different physical structures which are barriers towards getting your gene into a cell. And then here in the middle, you've got this structure called the nucleus. That's where we need our gene to go. Okay, we need our gene, our therapeutic gene, to end up in this nucleus in order for it to do its job. So the gene has got to go all the way from the outside, through the cell wall, through all of these structures, through this membrane that encapsulates the nucleus, and find its way into the middle. And that is not an easy job to do. It's really difficult to do. And so for that reason, we've basically hijacked viruses. Now I know that talking about viruses in the current global climate that we're currently facing in the COVID era that we're currently functioning in now is a bit tricky. But the reason why we use viruses is because getting genes into cells is basically what they do for a living. They are right at the top of the food chain of their ability to do that. And so we're kind of using them as a Trojan horse. We want to take a virus like this, take out all the bad bits in this virus and replace it with good bits. And the good bits that I'm talking about is, for example, the PLA2G6 gene, a fully functional version of the PLA2G6 gene. And then we can use this virus that's been modified to carry the PLA2G6 gene to deliver our gene of interest into cells of our bodies. Okay. So this is what a normal virus on the left looks like, and this is what our virus carrying the PLA2G6 gene looks like. Now, looking at them left and right, they look exactly the same. And that's because they are exactly the same from the outside. It's the inside that looks different because we've taken all the bad bits out, we've stripped them all out, and we've put the PLA2G6 gene in. So we've now ended up with this on the right. And that's taken us a while to build, but we now have a virus which carries that PLA2G6 gene. And that's great, but to be able to test this gene therapy using this viral vector that we've made, we need to have a mouse model. And we need to test our gene therapy to see if it makes a difference to the way the disease progresses in that mouse model. And we're lucky in a disease like INAD because there are a couple of mouse models available. The one that we've chosen to use carries something called a missense point mutation. And what that means is there's a tiny change in that gene that causes the function to be disrupted. It's the same kind of change that occurs in the vast majority of INAD patients. And that's why we like using this particular mouse model. The other reason why we like using this mouse model is because it does show all the problems that human patients show, and it develops quite quickly. Okay? So you basically have to cull this animal at 14 weeks of age because its disease severity has already reached its maximum point. And so we need to stop the experiment in those animals. So it moves quite quickly, which means that we can do our experiments more quickly. And it also has the same neurodegeneration that you see in patients in this mouse. So it has the same coordination problems, it has the same motor function problems, and it has the same neurodegeneration problems in the nervous system that we see in human beings. And that's a really important thing for us to have in this mouse model. It has to be able to recapitulate what's seen in the human form, so that this is a realistic evaluation of the gene therapy. And at the bottom here is just a picture. If you like pictures, which I like, you can see a picture of the gene here. And this arrow here shows where the mutation in the mouse gene is. So if you look at this mouse model, here, if we monitor the weights of these mice as they age, in red, you've got what normal mice look like. In blue, you've got what they call heterozygotes. It's if you have a mutation in one allele but not the other, they're normal. And that's okay, because that's what happens in patients as well. If you've got a mutation in one allele, the patients are normal. But if you've got a mutation in both alleles of the PLA2G6 gene, you can see the weights of these mice start to level off and drop off over time. And if you look at what these mice look like, on the right-hand side, you've got a picture of what a normal mouse looks like at 14 weeks. And on the left, you've got a picture of what a mutant mouse looks like. And you can see already the back of this mouse looks very different from what a normal mouse looks like. You know, they've got a lot of muscle wastage here, and they're hunched, and it's a kind of classic kind of sign in these mouse models of what neurological problems look like. At the bottom here, you've got a picture of survival. So the red line is the survival of a normal mouse, and that line goes on and on and on. But the mutant mouse needs to be culled at about 14 weeks of age because it has this really severe neurodegeneration. We can't keep them for any longer than that. It's the point that they die at. So what we've done is we've given these mice, these mutant mice, gene therapy. And we've looked to see, does it increase the lifespan of these mice? Does it improve the movement of these mice? Does it improve the way that cells in the brain are preserved? So we've tried to look at all these questions when we've given this viral gene therapy to these mice. And this graph here that I'm pointing my arrow towards is quite interesting, because where I have my arrow now is where the mice die if you don't give them therapy, okay? But all these lines to the right represent an increase in the lifespan of mice that have been given gene therapy in different kind of ways. So that's a really good sign, okay? They're all living longer. And in many cases, significantly longer than untreated mice. That's a big shift out to the right-hand side in terms of lifespan. If you look at how these animals move, so this box here represents the movement of a mouse. Imagine if you took a GPS tracker and you put it onto the back of a mouse. What we're doing here is we're looking to see how the mice move around when they're treated. So here in the middle, we've got a mouse that isn't treated, and this represents the end stage of this mouse. And you can see they can barely move, okay? They've got really severe neurological problems. Their back legs don't work. They're almost paralyzed in the back legs, and that's why they don't move much at all. On the left of this, you can see what a normal mouse looks like, and you can see mice run around just like normal mice do. That's what they do for a living. And then this box, and this box represent mice that have received the most successful form of gene therapy. And you can see that the movement of these mice is much more similar to a normal mouse than it is to an untreated mouse. So these mice are definitely moving around more and more active than untreated mice. And this is just from one treatment, right? This is just one injection of gene therapy when these mice are born. That's it. It's a one-off gene therapy. You don't have to keep giving it all the time. And then if you like numbers, then these numbers here, this graph here, represent what's shown in these boxes. So in red here, you've got what gene therapy treated mice, in red here, you've got what normal mice look like. In green here, you've got what untreated mice look like. So you can see they can hardly move. And up here, you've got the mice which have been treated with the most successful forms of gene therapy. That's great. Increase in lifespan, increase in their ability to move. Now, what I want to show you is a video. So you can see what we see in the cages of these mice when we treat them. So on the right, you can see mice which haven't been treated with gene therapy, and you can see they don't move much. They're having real problems moving around. They can't move their back legs. They've got neurological problems. And so they're sticking together and they'll stay there because they cannot move anymore. If you compare that to the left, the gene therapy treated mice on the left, these guys are moving around much more freely. They're able to move. You can see they're able to put their front paws up onto the side of the cages. That's called the rearing activity. They are far, far more able to move around, to have normal function, normal what we call locomotive function. So I'll just let that video move on for a few more seconds so you can appreciate how much more activity there are in the gene therapy treated mice. So one of the other things that we looked at was, is the gene therapy able to stop all those really important cells in your brain from dying? Okay. So we've looked in 3 parts of the brain here in these mice which are very affected. So we looked in the part of the brain called the cortex, the thalamus, and the brainstem. And what all these dots are showing is the number of cells that are present when we count them. So in the red, in each of these graphs in different parts of the brain, you've got what the normal number of cells look like. In green, in different parts of the brain, you can see what the cell count is in mice that we haven't treated. And you can see the number of cells have really come down. You can see the big difference between the green dots and the red dots, because those cells are dying. But when you give the gene therapy in its most successful form, so the black or the blue or the purple, you can see that number shoots up. And that's because the gene therapy is preserving those cells, it's protecting those cells from dying off. And you can see that's the case in all 3 parts of the brain. And even when you look in the spine, when you look at different cells that are in your back that run down, are important in connecting the central nervous system with other parts of the nervous system called the peripheral nervous system, which is important in this disease. In red, you've got what normal mouse counts look like. In green, you've got what untreated mice look like. And in black, you can see what the gene therapy does. So here, in one part of the spine cells, black cells are almost completely normalized back to normal levels. And another part of the spine, you can see that's the case as well. So it's fantastic. You're having a neuroprotective effect in the brain, and you're having a neuroprotective effect in the spine. Now, there's a lot of kind of impetus building in gene therapy for neurological diseases at the moment. And much of this is being driven by clinical successes, and not just clinical, but commercial successes as well. So there's now been a licensed gene therapy for another pediatric neurological disease called spinal muscular atrophy. And this is great news for the gene therapy field, because this is a gene therapy that's gone from what I'm describing to you in mice, has gone to human beings, and has gone through the commercialization process, which means that there is now a licensed drug for this awful condition called spinal muscular atrophy. And so there is this is kind of giving us a lot of hope in the neurological field for progressing these gene therapies into a real commercialized, meaningful drug. And this is the model that we really want to try and achieve in terms of gene therapy for INAD. And of course, this isn't the only gene therapy trial, they're a licensed product. There are other trials taking place. This is a clinical trial for a disease called Batten's disease. You may have heard of it, you may not have heard of it, but it's another rare neurological pediatric condition. There's a gene therapy clinical trial running for that as well, and you can look this gene therapy clinical trial up. It's publicly available, you can see the details. And so this is all great, and we're learning a lot, but we also don't know a lot. And that's because this is a relatively new field. And that's because these clinical trials are still running. Okay, so it's great they're happening, and we need to learn from them, but they're still ongoing, and we don't know what the end stage of these ongoing clinical trials are going to be. But we are hopeful, and we are all, I think, everyone working in the gene therapy field, very enthused by what we're seeing at the moment. So I think I'll stop there. Professor Curran is going to tell you more about the next steps that we are looking at, and she's going to tell you about how she and her colleagues are ramping this up. But just to say thank you to all of our colleagues here at UCL, where we've been developing this program, and also to Ken Seino's lab in Japan for supplying us with the mouse model that we've been using. So I'll stop there, and I will hand back to Manju. Thank you very much.
Thank you, Ahad, for a wonderful introduction into gene therapy and how we're planning to apply it to INAD. Really lovely overview and some very beautiful results. There's lots of questions on the chat. We have a few minutes of questions. So I'm just going to combine a couple of questions from Anna Messias. She's asking, when did you perform the gene therapy? How old are the mice? And if the gene therapy is not applied at an early age, are the damaging effects in development reversed?
Yep, thank you, Manju. So those are very nice questions from everybody. So what we're doing at the moment is that we're performing the gene therapy very early on. So when the mice are born, we're giving them at that age. And I think the questions that are coming through as to what happens if you are to treat the animals a little bit later. So for example, when they're early symptomatic, which would realistically mimic the clinical reality of the situation upon diagnosis, is a very good question. And those studies are being performed at the moment. So we have a grant from the Medical Research Council, which is allowing us to fund us to be able to perform those critical experiments. My feeling is that if we are able to treat those neurons which are there, there's a chance we can protect them if they're healthy. But even those neurons which are on the path of degenerating away, if we're able to catch them at a certain point along that timeline, we may be able to even rescue some of those neurons which have started that process. So it's a matter of getting in there as early as possible. And that's why I think the biomarker studies and the diagnostics involved in this disease are absolutely critical to allow us to intervene as soon as possible.
And then Hein Sherpas, have you also seen adverse effects from too high expression?
Yeah, thank you for the question. That's critically important. So I haven't shown the data here in the interest of time. We've injected incredibly high doses of our viral vector into the brains of these mice and overexpressed to massive levels PLA2G6. And we have looked at neuron counts, we've looked at microglia-mediated inflammatory response, and we've looked at astrocyte-mediated inflammatory response, and we have not seen any signs of toxicity associated to overexpression of PLA2G6 in the mouse model. This, of course, requires to be repeated at a much, much higher level by an independent laboratory from us, but our initial studies are showing no problems with overexpression.
Odie Sabon, what is exactly representing the grey line in your graphs referred to as AAV9 HPLA2G6 IV? So this is not so successful. So can you explain why?
Yes, it's a very, very, very astute observation. So the AAV9 vector can be given via multiple routes actually. So one way to do it is to inject directly into the nervous system, which is what we are doing. But AAV9 also has the ability to cross the blood-brain barrier as well. And so we were at the beginning wondering what is the best way to do it. Do we inject it directly into the brain, or do we inject it intravenously? So inject it into a vein, for example, which would be much less invasive and much more convenient for patients. The thing is that route of administration, the intravenous route of administration, was not very successful. And so this is the reason why we do these experiments and we keep all options open, so that we can try and dissect out these things in a very transparent manner. And so our experiments are showing that actually the intravenous route of administration for this particular viral gene therapy is not the way forward, and that the direct injection into the cerebrospinal fluid in this mouse model and in our hands is a better way forward for translation to potentially human beings.
Thank you, Ahad. I think in the interest of time, there are a couple more questions, but may I ask you to answer them on the chat, if that's okay, so we can keep to time. Thank you for an excellent presentation, and we'll continue.